So in the second part of discussing synapses, I'm going to go through the chemical events or what is happening at the synapse, as well as to start talking about the neurotransmitters or the chemicals involved. So I'm going to jump back into the history of this for just a minute as we talk about this chemical transmission that's occurring at the synapses. If you remembered, so it was Charles Sherrington who inferred the properties of a synapse and noticed that there had to be this neural communication, that what was happening in the reflex arc was too slow to be just conduction along an axon. But he believed that it still was occurring too quickly for this to be a chemical transmission. He believed that it was an electrical communication at the synapse as well. Other people were looking into this idea of chemical transmission, and one of those people was T.R. Eliot. And in 1905, he applied adrenaline directly to the heart, stomach, and pupils, and noted that this had the same effect as activating the sympathetic nervous system. So increase in heart rate, slowing of digestion, and dilation of the pupils. And so it looked like that the sympathetic nervous system was causing these results by releasing adrenaline or some similar kind of chemical. However, Charles Sherrington had a lot of prestige and a lot of respect, and it could be that this applying adrenaline was just mimicking the effect of the sympathetic nervous system, but not indicating how the sympathetic nervous system worked. So people did not believe from Eliot's work that this was a chemical transmission. Now in 1920, Otto Loewi did this study where he repeatedly stimulated a frog's vagus nerve, so lowering the heart rate of the frog. And then he collected a fluid the heart was in this fluid. He collected the fluid from around the heart and transferred it to another frog's heart. And what he found was that it caused the other frog's heart to also decrease its heart rate. Okay, he also did this with increasing heart rate, so stimulating the accelerator nerve, increasing heart rate, collecting that fluid, transferring it to another frog's heart, and causing an increase in the other frog's heart rate. This really strongly suggests that this is chemicals and not electricity. He's not pulling out from the fluid loose electricity. There are chemicals in that fluid. I love the story of Otto Loewi. He actually had this dream where he came up with this idea, woke up in the middle of the night, wrote himself some note, woke up the next day and looked at his note, and he wrote it at 3 o'clock in the morning, so he couldn't read his own writing. Woke up the next night having this dream of how to look for chemical transmission, and ran to his lab. Woke up in the middle of the night, ran to his lab to go start this study. He said in the light of day he didn't believe it would work, but in his dream mind it seemed like it would work, and it did. And so now people really did believe this is a chemical transmission that's occurring at the synapse. Okay, so by the 1950s, researchers had established that chemical transmission is really predominating throughout the nervous system. We have a few electrical synapses that we'll talk about really briefly towards the end of discussing synapses. But this idea that it's a chemical transmission really revolutionized our understanding of the nervous system. And if you know your bit of history about how these kind of psychotropic drugs, or how we came up with these psychiatric drugs for people, this was happening in the 1950s. So this really revolutionized the way we treat people with any kind of mental disability or mental illness. So we see this development of psychiatric drugs. I have the example of chlorpromazine, which came out to help people with schizophrenia, and this really kind of emptied out some of those institutions. Not emptied out, but it cleared out a lot of people who were helped enough by chlorpromazine that they could at least handle the real world again. So I'm going to go through some of my basic terminology, which up to this point it might have sounded like I already assumed you knew a bit, but before we talk about synapses in more detail, I think it's really important to make sure we're all on the same page. So that synapse is also called a synaptic cleft. That is the microscopic gap between the terminal buttons of a ascending neuron and the cell membrane of another neuron. So whether that's at the soma or more likely at the dendrites. When the action potential reaches those terminal buttons, it triggers the release of these chemicals, or the neurotransmitters. Those neurotransmitters are stored in what are called synaptic vesicles, sometimes called terminal buttons, or boutons, the French for button. So you'll see all of those, sometimes called synaptic sacs, S-A-C-S. So you'll see all of those in different kinds of literature and throughout the book. Sometimes he goes back and forth, as many people do. The action potential, when it gets to those terminal buttons, it causes the synaptic vesicles to fuse with the membrane at the end of the axon and then spill those contents into the synaptic cleft. And here I'm going to walk you through some pictures with a lot of what I've said, but I do want to make sure I'm using the terminology and saying all the words that I need to say. I've probably gone back and forth a couple of times now between saying the sending neuron and the presynaptic neuron, and then the receiving neuron and the postsynaptic neuron, and I've tried to do that together so that you're putting those together in your mind. And then, so when I'm a postsynaptic neuron and I'm receiving the signal, remember, I'm probably sending that signal on somewhere. This is the point, it's neural communication. So in the next, it's really this cycle, so that in the next line of communication, I'm going to be the presynaptic neuron, and the next neurons down the line are going to be the postsynaptic neurons, the ones receiving my signal. So we have those neurotransmitters, those synaptic vesicles fuse onto the membrane. They spill those chemicals, those neurotransmitters, into the synaptic cleft. Those neurotransmitters diffuse across the synaptic cleft to the membrane at various receptor sites or binding sites on the postsynaptic neuron. Again, mostly on those dendrites. Receptor sites are really tuned to recognize certain neurotransmitters and not other neurotransmitters, so they usually use this lock and key analogy. So if I'm spilling a particular neurotransmitter into a synaptic cleft and the postsynaptic neuron doesn't have any receptor sites that recognize that neurotransmitter just does not have an effect. Okay, that we need those receptor sites that are tuned to recognize where the specific neurotransmitter will fit inside those receptor sites, kind of like a key. So I'm going to walk through this sequence of chemical events at the synapse, which I've already done some of it with the language above. What I have here on this slide is, on the left is a picture from one of my Psych 101 textbooks that I've used in the past, and it has been my favorite picture. I've kept it and used it in many, many classes. It is a pretty simple description of what gets a little bit more complicated here as we get into the PSYC460 level of talking about these things. But if you ever want to go back and just kind of look at the simple, oh yes, this is my kind of skeleton and what I need to know, you might want to look back at that. So for me, this figure on the right is really difficult to see, and I don't know if that's true for you, but it is in your textbook on page 51 if you want to look at that as I walk you through this. What this is showing overall is the presynaptic neuron, that terminal button, one of the terminal buttons from the presynaptic neuron, and we can see that the neurotransmitters are synthesized, and in those synaptic vesicles there's little round balls in the terminal button. And then on the other side, so we have the synaptic cleft, that space between the two neurons. On the other side, we see the postsynaptic neuron with its receptor sites, and so that's probably a dendrite or a dendritic spine, some piece there. And then around that is the glial cell. If you remember, the glial cells are really helping to isolate those synapses, keeping out the other ions that might be floating around in our extracellular fluid, so that this what's important here is the neural communication between these two neurons. Okay, so I'm going to go through these numbers 1 through 7 of the sequence of events. So very first, we have the synthesis of smaller neurotransmitters. He gives the example of acetylcholine. That's occurring here in the terminal button. Some of these vesicles, we have some synthesis up in the cell body, and they travel down. but some of them are synthesized here. The action potential, if we send an action potential in the presynaptic neuron, it's going to eventually get to this terminal button, and it's going to trigger the release of neurotransmitter. And it does this as it gets here. It allows calcium to enter, which then causes an exocytosis. So exo leaving, CYT, the leaving of the cell. So we see a release of neurotransmitter. That's in number 2. In number 3, so what then happens is the neurotransmitter diffuses across the synaptic cleft. And if you look at number 3 there, that is the neurotransmitter binding to the receptor sites. And remember, those receptor sites have to recognize that particular neurotransmitter for the neurotransmitter to fit in and to bind there. Number 4 is the separation of the neurotransmitter from those receptors. And so it's going to go back into that synaptic cleft. And a number of things can happen to it here. It can kind of diffuse away. It can be taken back up by the presynaptic neuron in this process of reuptake. It can also be broken down by enzymes, and I'll go through those steps. But so in this picture, number 5 is this process of reuptake. The presynaptic neuron is pulling back up those neurotransmitters that are in the synaptic cleft. In number 6 here, the postsynaptic cell also releases a retrograde transmitter, so that it's going to slow or inhibit further communication from the presynaptic neuron. I usually compare this in a very colloquial kind of way of, if I keep sending you an email and I'm telling you the same thing over and over again, you might send me an email saying, I've got your message. You don't need to send that email again. Okay, this is just letting the presynaptic neuron know, I've gotten a message. You can slow down your communication with me. And then in number 7, we also have these negative feedback sites that are going to respond to that retrograde transmitter. And sometimes they respond also, they have these autoreceptors to my own. So as a presynaptic neuron, I can release these neurotransmitters. They get into the synaptic cleft and they come back up and fit into my autoreceptors, basically like me. When I send you guys email through the Blackboard system, I send that email to myself, again, letting me know I just sent this message. So that's the presynaptic neuron really letting itself know you just sent this message. So both of those things really inhibit further communication. Okay, so I always feel like that's quite a bit to do with a picture, and you, of course, can ask questions if you have them. I know that there are also descriptions on YouTube that basically go through a lot of what I just went through, but with animation, so it's a little bit more fun if you want to look for some of those or ask me about some of those. But do think about asking questions. I'm going to walk you back through all of those 7, that sequence of events, those 7 steps, but with the language on the slides instead of looking at the picture. So this is repeat, and again, you can think through if you have questions. So the major sequence of events that allow for this communication to occur between neurons. In the presynaptic neuron, we have synthesized chemicals, or neurotransmitters. They're stored in those synaptic vesicles or those synaptic sacs. The action potential, if I'm going to send this information on, the action potential is traveling down the axon, okay, and reaching that terminal button. When that action potential depolarizes that area in the terminal button, the membrane of the terminal button, this opens calcium channels, which causes exocytosis, so that leaving from the cell, the neurotransmitter is leaving from the cell. And I like the picture that's on the left there that is showing the membrane before the action potential gets to it. And you can see there are these little dots that are almost impossible to see on my screen. But then if you look down below it in B, those little dots have gotten larger, and that is where those synaptic vesicles are fusing onto the membrane, and they're going to be dropping those neurotransmitters, or those chemicals, into the synaptic cleft. Those released molecules, those neurotransmitters, attach to receptors on the postsynaptic neuron, and alter the activity of the postsynaptic neuron. And remember, this is where we can see this can be excitatory, this can be inhibitory, depending on the neurotransmitter and the area of the brain and all of that. So it's altering the activity and altering the likelihood that that postsynaptic neuron will fire. That then, number 4, the neurotransmitter molecules separate from their receptors. Okay, so if the neurotransmitters linger at that postsynaptic membrane, they can continue exciting, inhibiting the postsynaptic cell, but having whatever influence they have, they'll continue altering the activity of the postsynaptic cell. So we really want to get those neurochemicals out of that synapse. And a number of outcomes for neurochemicals are possible. If you noticed, number 5 in the picture was the process of reuptake, but I start here with the one of the ways of getting those neurochemicals out of that synaptic cleft is when they are broken down by enzymes, which is basically a recycling system. I'm going to give you this example, and then I'm going to go through my story that I think stories help us to understand and really remember things, so I'm hoping this story helps and it's at least interesting. So the example of acetylcholine. So acetylcholine is one of our neurotransmitters. It's going to activate the receptor, have its effect on the postsynaptic neuron, and then it's going to come back out into the synaptic cleft and be broken down by this enzyme, acetylcholinesterase. It gets broken down into 2 fragments, acetate and choline. The presynaptic neuron takes up the choline, and it has to reconnect it then with the acetate, so it synthesizes the acetylcholine there in the terminal button, and so that we can send acetylcholine again. Okay, so to help you remember some of this, I have this example of myasthenia gravis, which basically means grave muscle weakness. This example comes from Neil Carlson, one of the textbooks I've taught from. He started his chapters with these vignettes, with these real life applications to what he was going to talk about. And this one was, I thought, a really interesting one. So Catherine is, her supervisor has just left, and she starts taking over some of his responsibilities. She's one of those really go-getter, active people. So after work every day, she goes to her aerobics class, then she goes swimming, and then she goes home. What she starts to notice first is that she can't make it through the aerobics routines. She can't make it through class. She has to take breaks. And she gets tired when she's swimming and doesn't swim for as long. Pretty soon, she has to stop doing the aerobics class. She thinks this might have something to do with stress and everything going on at work, but she decides to go see her doctor. And her doctor listens to the symptoms, listens to her what's going on at work. You know, that she's got all these new responsibilities. People are watching her. She might be promoted. There's just a lot more stress. And the doctor says maybe it's just stress, and we'll see what happens down the road. And if this continues or gets worse, you can let me know. So she goes back to work. Time passes. She eventually she starts doing an easier stroke when she's swimming. Pretty soon she's not swimming very long at all. Pretty soon she stops her swimming too. So now she's not doing the aerobics or the swimming, and it's really even beginning to affect her work. And she decides to go back to the doctor again when one day she's at work and she's working on something, and she looks up at the clock and she realizes that her eye is drooping. She feels so fatigued, and she thinks this fatigue is so intense that she can't even completely open her eyes to look up at the clock. So she gets really concerned at that point, and she goes to see her doctor again. And the doctor is noticing that this is pretty severe, and so she suggests to Catherine to go see a neurologist. Catherine sees the neurologist, describes the symptoms, describes what's going on at work, but then the neurologist says, well, let me try something. And Catherine gets an injection, and she starts to feel better. She can already feel her energy coming back. She's like, I feel like I could go back to work and work now. I feel I'm completely better. This is weird. Okay, and the neurologist tells her, well, what I did was inject you with this physostigmine, this medicine that basically breaks down acetylcholinesterase so that you don't have that enzyme in your system. Because what's happening in myasthenia gravis, she has this genetic disorder, which is really quite rare, where her immune system starts to attack the acetylcholine, that particular neurotransmitter. So she has so little of it that when it's getting broken down, she can't even move her muscles. This is actually going from our motor neurons to our muscles, in addition to being in various areas of the brain. So from then on, she can take a pill that just basically destroys the acetylcholinesterase, which allows that acetylcholine to stay in the postsynaptic or in that synaptic cleft for longer, so that it can alter the postsynaptic cell. So hopefully that was helpful, or at least an interesting story for people. So this being broken down by enzymes or enzymatic degradation, this is a recycling system. And just like recycling in the real world, it takes time. In this case, not every molecule is reabsorbed. And if action potentials are coming fast enough, they can deplete these neurotransmitters faster than the presynaptic cell can replenish them, which can slow or interrupt transmission. Okay, so the second outcome for these neurochemicals after they influence their receptor sites is that they can be taken back up by the presynaptic neuron in this process of reuptake. Serotonin and the catecholamines, so dopamine, epinephrine, and norepinephrine, they are not broken down into inactive fragments in the extracellular fluid. And that what happens instead is the presynaptic neuron is going to take up most of that released neurotransmitter, and then it puts them back into synaptic vesicles so that they can be reused. And you can see that this is a bit more efficient than that recycling system. It's not taking as much time. We're not losing as many molecules. This process of reuptake occurs through special membranes that are called protein transporters, and people differ in the activity of certain transporters. And so one of the examples that we see is differences in the serotonin transporters, these genetic differences where different people have different likelihoods of having anxiety, post-traumatic stress disorder, depression, depending on whether they get what's called the long serotonin transporter or the short serotonin transporter. And actually there's the two alleles, the one from their mother and the one from their father. So if I get two longs, I can go through some pretty severe traumas and not have that come out as post-traumatic stress disorder or some kind of phobia or anxiety or depression. If I get a short and a long, kind of more likely, but I can still go through a number of pretty traumatic experiences and be okay. If I get two short serotonin transporters, it looks like I'm pretty likely to, if I'm going also through some traumatic experiences, I'm more likely to now show anxiety in response to certain things, or post-traumatic stress disorder, or depression. Transporters also differ in their abundance in different brain regions, so from one brain area to another. And I just have an example here, although it doesn't mean a lot now, but it will later. The dopamine transporters in the caudate nucleus, we have different numbers of dopamine transporters in the caudate nucleus, in that subcortical structure, than we do, let's say, in the prefrontal cortex. And finally, the third outcome for neurotransmitters after they influence their receptor sites is that they can just diffuse away. Okay, they just kind of go off into the extracellular fluid. And again, going back to that picture from an earlier slide, or from page 51 of your book, numbers 6 and 7 have to do with this negative feedback, really inhibiting further synthesis and release from the presynaptic neuron. So number 6 is that the postsynaptic cell can send back reverse messages to slow or inhibit further neurotransmitter release by the presynaptic neurons. your author gives this example of nitric oxide. Nitric oxide is actually a gas, but this is a pretty common neurochemical. I sometimes go back and forth, and I really try to use the word neurochemical because some of these are not exactly neurotransmitters, and but a lot of authors just call them neurotransmitters. So I apologize that I forget. But so this gas that doesn't actually need a receptor site, but there are other possible neurotransmitters that can then be sent from the postsynaptic cell back to the presynaptic cell to slow release or slow communication. There are also, number 7, receptor sites on the presynaptic terminals that respond to those neurotransmitters that are released by the postsynaptic neuron. And we also have receptor sites that respond to my own neurotransmitter release. So the presynaptic neuron releases its neurotransmitters, and they can go in and fit in my own receptors, the presynaptic neurons receptors, called autoreceptors, is AUTO, right? Is auto is myself. I'm recognizing my own receptors, and this too will inhibit further synthesis and release. This kind of negative feedback system.